Hello, welcome you all. Today we are going to see animal cell culture media. Animal cell culture, as far as we have seen in our syllabus, the culture of animal cells. We have learned a little bit about a historical uh, segment of animal cell culture. Then we have learned several things about the terminologies in animal tissue culture media or animal tissue culture subject. We have also seen several topics in terms of cell culture and its usage, like the application of animal cell culture. Now, today we are going to see what kind of media that we generally use in animal cell culture. If you see the cell culture based on its nutrition, we generally devise its media. The idea of cell culture was came up from the blood cells actually, because blood contains several cells. You all know that hematopoietic stem cells differentiate several different types of cells in our blood, and then ultimately these cells they suspended in the blood, and the plasma or serum is itself provide nutrition or nutritional factors, carbohydrates, proteins to that particular cells. Now if you see in the animal tissue culture media, we have seen a little bit about applications also. In applications, we saw that what are different applications in animal cell culture. First of all, we generally do tissue applications for studies in intracellular activity. This is a kind of revision, a little bit revision. Then the to understand hormonal receptors, how exactly hormones works, what is the uh, usage or what is the application of signal transduction, different mechanisms which is going on inside the cells and outside on the surface of the cell. Then to study cell adhesion molecules, CAM molecules, motility and metabolic cooperation of cells. Then we need to understand cytotoxic effect of different chemicals, different drugs, toxic elements or toxic compounds on cellular level. We need to do a mutagenesis also. We generally genetically modify say several cells or cell lines, and we do check the impact of environmental impact or interactions of different compounds with these genetically modified cells or transgenic cell lines. That kind of study also we do with the help of tissue culture. And then genetic analysis, immortalization, senescence. Immortalization means those cells we generally transform them into infinite cell culture. That culture we generally study. Then laboratory productions of different medical or pharmaceutical compounds, and the wide range of applications like vaccines, interferons, hormones. So such kinds of compounds that we generally produce from animal cell culture. Now if you see the animal cell culture are actually useful for this wide range of pharmaceutically ingredients or pharmaceutical different compounds. So what kind of compounds they contains? They are basically proteins, molecules or segments of peptides which are helpful in reducing several symptoms of any disorder or disease, or to cure that particular disease or disorder. Example is plasminogens, interferon, then blood clotting factors, hormones, monoclonal antibodies, then erythropoietin. There are several types of different factors of erythropoietins are there. Then in addition there are like interleukins are there. um, TNF alpha, like TNF beta, such a kinds of factors are also very important. They are kind of chemokines or cytokines which are really helpful in immune responses. And so pharmaceutically or medically they are really very potent or important to cure or to suppress the activity of any foreign antigen or foreign any bacteria or viruses. So the major development in cell culture technology is like development of, first of all, antibiotics is the major development, guys, because this is very important, because antibiotics in the cell culture media leads to um, the like it inhibits the growth of other bacteria, or it inhibits the growth of other contaminants. And so after all, it inhibits the growth of contaminants, and so that leads to pure culture. The output is very important. First of all, we need to understand over here, uh, animal cell culture is the process which is been conducted in a very aseptic condition. Very sterile environment is necessary to perform this activity. So the contamination leads to malfunctioning of cells, or variations in developmental process, variations in metabolic activity of any particular cell line, and that leads to false production of any undeserved component like proteins, fatty acids, or any other extracellular components that is necessary for our study. The second, so with the use of these antibiotics, generally uh, we generally perform and tissue culture techniques, and we get a very pure culture out of it. Second was the use of trypsin. So basically trypsin is an enzyme. It's a class of protease enzyme which cleaves protein molecules. So what happen, cell when there is a adherent cell culture, then these adherent cell cultures are like the cells which interact with the other cells, and they form a linkage, or they form bonds in between them. And these bonds or these linkages is with the help of receptors which are present, or several anchored elements, anchored protein which are present on surface of the cell, and they engulf to each other, and within that engulfing they attach to each other, and ultimately we call them as adherent cell lines. These trypsin enzyme or trypsin protease enzyme, it is useful to cleave these adherents or to cleave these bondings which is present in cell junction. And so that leads to production of completely a free or suspended cell culture, so that we can subculture them, we can propagate them in the other flask. So these are wide applications of trypsin, which is really helpful in animal cell culture. We will see more about trypsin in our next lecture when we learn about subcultures in detail. The third development was chemically defined culture medium. Today we are going to see the media only, so we are going to see what is the chemically defined media. In other terms, it is also called it as serum free media. Okay, we'll move to next slide. Then what is culture medium? So the culture medium is means a growth of medium or culture medium is the liquid or gel designed to support the growth of the cell. So this is really very basic definitions. In other terms, we can say that the culture media is like which provides nutrition, which provides the suitable environment to the culture or the cells, animal cells, and that all these things, when a single chemical composition, when it provides, that's what we call it as culture medium. So never written a bookish definition at all, because whatever you have understood throughout your throughout your understandings of animal cell culture, you just write down according to your understanding. uh, Different types of culture medias are available. The first is natural media, and the second one is artificial media. If you see the natural media, in the early years, the natural media obtained from various biological sources. These biological sources, biological fluids, if we count it in terms of like plasma, serum, lymph, human placental cord serum, then amniotic fluids. So these are a few biological fluids which has been extracted from humans or any other mammals or animals, and we were using these fluids in culturing the animal cells. So that was the previous part, and that is also called it as natural media. Then the next thing that is been used in natural media are tissue extracts. Now guys, tissue extract means what exactly? Whatever tissue that we excise or we just cut down from the uh, mass of any animals or from any animal organism or organ, we used to squeeze it, or we used to grind it in a grinder, and then ultimately whatever extraction after the centrifugation we got it, it's called it as tissue extracts basically. So it's amongst the tissue extract, cheek embryo extract is been used widely. It is the cheek embryo extract is the most commonly employed media in the natural media. The extract of liver, spleen, bone marrow, tumors, leukocytes, then the bovine embryo, and almost like I said, chick embryo. So these are few things, tissue extract that we have been used in natural media. The third one is the clots. The clots means the coagulants or plasma clots basically. Uh, Now if you see what is mean by clot means something which has been grouped. Like in our body, there are like few proteins present in our blood like fibrinogens, factor 8, and these are responsible for clotting. They helps in clotting of blood, because to reduce the injury from the injured part or from the wound which is happen on the exterior epidermal layer. And so these coagulants and these clots, they form because they form a mesh like structure. When they form a mesh like structure, um, they actually can if you see the artificial media, then you will find the artificial media have been in use for cell culture since 1950. The minimal criteria needed for choosing a medium for animal cell culture is like, take example of the media should provide all the nutrients to the cells, then it should maintain the physiological pH around the like 7, with the adequate buffering system. Then the third criteria is like the media must be sterile and isotonic to the cells, because you all know that what is meant by hypertonic, isotonic, and hypotonic. So isotonic gives the best result, best buffered solution to the cell, so cell will not get any hampered due to any salt solutions, excessive or less. So this is the basic criteria for artificial media. If you see the example, then you can see that balanced salt solution is one of the artificial media which has been used in animal cell culture widely. And the examples are like Eagle's basal salt solution, Hank's basal salt solutions, and these are few salt solutions. um, and then if you see that the basal, in the basal media there are like the another media, they're like MEM, like modified Eagles medium, or Delbecque's modified Eagles medium. So these are few like examples of basal media. The fourth one is complex media, and within that complex media, there is a mixture of buffered solution as well as nutritional excessive factors, and we can change or we can manipulate these uh, media compositions with respective or with the requirement of our cell culture preparation. The examples are like RPMI 1640, then IMDM media. So these are few examples. Uh, these are the two major types of tissue culture media. You should know this because these are very important while writing your examinations. If you think about natural media, then natural media is very useful if you see the application. But the one drawback is that lack of knowledge of exact compositions of these natural media, because if you say so, we are using any egg extract or any blood extract like serum or plasma, there whatever source that we have taken like bovine serum albumin or bovine serum that we are using, so we exactly don't know what is the proportion of cellular growth factors, or what could be, or what kind of cellular growth factors are there inside the that serum or plasma media. So according to that, our cell cultures might get deviate or might get false regulations, and that leads to abnormal development or abnormal secretion of any extracellular chemicals. So that could be done, or that has been done based on this natural media. So up to the educational level or up to the teaching level, this natural media is okay. But when you do such a research on particular cell lines, and you are observing particular signal transduction pathway, or you are observing mutagenic any effect or the mutational expression of any genes on cells, and you are observing the extracellular component which has been secreted from particular cell, at that time you should use artificial media, because the intake you should know. So and so only you can devise or you can formulate the output of the cells or output of your cell lines. Then we'll shift towards artificial media. If you see the artificial media, the artificial media are like serum containing media, yes, serum free media, then chemically defined media, then protein free media. So these four medias are there. Serum containing media, of course, we generally use 5% to up to the 20% of serum that we generally use in the media, and that is based on completely types of different cell lines or types of cells that we are culturing in. Next one is serum free media. In the serum free media, defined culture media is there. As far as I just told you a little bit information in artificial media, the defined culture media means we know what is the percentage of glutamine and what is the percentage or what is the content of what kind of growth factors are there, what kind of hormones are there, what is the proportion of fructose, glucose, any other carbohydrates than that of these monosaccharides. Then is there any addition complementary molecules are there or not, what is the buffer situations, what kind of osmoregulatory components that we have added inside the media, that all these things that we do with the help of defined culture media. Then chemically defined media is a little bit different, because in the chemically defined media, only and only inorganic things or inorganic components that has been used as a media, no any biological component like serum or any other tissue extract or any other thing that has been used. Then the protein free media, few cells are there where we generally understand particular cell lines, and we generally used to see the effect of different nutritional factors, different amino acids, and its impact on cell lines. The best example is that your exercise gym or your like there many products are there to the sellers, like body mass grow or mass grow or such a kinds of protein powders or such a products are there, right? Now these protein products, they contains high amounts of particular proteins or particular amino acids which regulates growth of the cells or the muscular growth pathways, like metabolic pathways. And in excess of this amino acid, the muscle cells leads to excessive cell division, or that leads to the growth, it enhances growth. So these products came into the market based on these understandings. What kind of amino acids provide what kind of metabolic pathway, or which amino acid enhances which metabolic pathway in a particular cell. So that study we generally do with the help of protein free media. Protein free media means we generally skip all the proteins out of it, and we generally add a specific amino acids or specific components that regulates the protein intake of cells, is it? So there we use protein free media. There are other uses are also there, but one example that I have mentioned here is one of the famous example, and you all know these things. We'll move to next slides. Basic components of cell culture. In the basic components of cell culture media, it is, they are available in the powder form also and the liquid form also, is it? So they are available in powder form and liquid form. In the powder form, the variety of different chemicals are continuously available. Some are in absence of vitamins, some comes with the presence of vitamins like biotin, then choline chloride, folic acid, inositol, nicotinamide, or pyridoxine HCl, or riboflavin, thiamine, vitamin B12. This variety of vitamins are there, and that comes up with a different variety. It depends on your research, or it depends on your work, that what kind of product that you have to choose. Then other components like glucose, phenol red, sodium pyruvate, lipoic acid, linoleic acid, hypoxanthine. These are variety of different components that are necessary. Some component, sorry, and some components are not necessary. So depends on again your work. So I need to tell you that what is exactly the, first of all we'll see what is the buffer, what is mean by buffer, because this is very very important thing, guys. This is very really really very important thing, because if you see the buffering in the cell culture, that leads to regulation of cellular osmosis, or that leads to regulation of the cellular impact or cellular system inside the tissue culture. Now what, how it works? So first of all, just take example, the reaction, is it? We'll take a reaction. If you say this is the CO2, right, the carbon dioxide. So basic reaction is that when carbon dioxide reacts with a water molecule, it form what it forms, it form a carbonic acid, H2CO3. It form a carbonic acid, and when that carbonic acid reacts with the proton which is available in the water, okay, carbonic acid, when it reacts with the proton, it again either turns it into carbonic acid, okay, or it form carbonate ion, CO3 minus ion, and then again H plus ion, the proton molecule. You got it? So this is basic reaction. I'll again teach you, just listen to me. Carbon dioxide in the medium is in the dissolved state, right? The carbon dioxide is in the dissolved state, the concentration of which depends on the atmospheric CO2 which is present, right? These are atmospheric pH CO2, or which is basically present in the media or in the incubator. And so CO2 in the media exists as a carbonic acid. Is it carbonic acid? And the other thing that we incorporate, is the bicarbonate. Okay, sodium bicarbonate we generally incorporate, NaHCO3, which is not mentioned over here, but I need to mention over here. um, Na2CO3. Na2CO3 leads to what? Na2CO3, that sorry that leads to the production of Na plus sodium ion and HCO3 minus. So HCO3 minus means this bicarbonate ion. Is it? So what exactly happened? As in the evidence from the above equation, like from the this above equation, the concentration of CO2, HCO3, and ultimately the pH are interrelated. By increasing the atmospheric CO2, if we increase the atmospheric CO2, the pH will be reduced, making the medium acidic. Are you getting my point? If you increase the CO2, it will interact with the water molecule, it will form carbonic acid. Is it carbonic acid? Then again turns it into, or it dissociates into two ionic form. One is HCO3 minus and H plus ions. H plus ions, in short terms we say that it's what? It's the proton molecule. And once the proton molecule increases, the acidity increases, and that lowers the pH. Is it? So the medium becomes acidic. If we add, now like just I said, if we add this Na2CO3, if we add this Na2, sorry, sorry, my mistake, NaHCO3, that is NaHCO3. If we add this NaH sodium bicarbonate, if we add this, is it, what happen? If we add this NaHCO3, it dissociates into sodium ion and carbonic ion or carbonate ion, bicarbonate and HCO3. So then what happened? The commercially available media contain generally these carbonate ions in presence, but sometimes if you yourself are going to formulate the media, then like you need to add sodium bicarbonate separately, and that has several percentage, like in 1 liter of media how much gram of sodium bicarbonate that you have to add in excess. The bicarbonate buffer is helpful because at the one side the bicarbonate ions increases, ultimately it associate with the H plus ions and become carbonic acid. Is it? And in other terms, other bicarbonic acid reacts with Na plus ion, sodium ion, and it becomes sodium bicarbonate, which again increases the pH. So at one end this chemical reaction is pulling the whole chemicals or the chemical properties towards the acidity, and in the other end the chemicals are pulling these reactions towards the basic property or basicity, because sodium bicarbonate is ultimately salt, which reduces the H plus ion concentration. So decrease in proton concentration leads to increase in pH. So tension this tension is there. So whatever excessive protons is being generated from the cellular mass, because you all know that cells, our metabolic process is contains respiration process, where high amount of proton flux is being generated in mitochondrial membrane, and that leads to excretion of this proton or highly acidic components into culture media also. So this culture media is ultimately the deposition of these extracellular acidic components into the culture media leads to decrease in pH, like the acidity is increases, and so there the sodium bicarbonate ions are there, and so these sodium bicarbonate ions, they are pulling these all chemical or media component towards the basic or towards the salt solution, and that's the very fantastic procedure which happens with this sodium bicarbonate system or carbonate bicarbonate buffer system. I hope so you got this thing. I'll post one animated video is also there. Okay, so next thing, if CO2 is important, so then what about oxygen, right? The great majority of cells in vivo are dependent on oxygen, because oxygen is the last electron acceptor in our respiratory mechanism. So that's why we need oxygen, and that is helpful in our aerobic respiration. You all know that, not anaerobic. Okay, anaerobic is glycolysis, if you know, say so. The aerobic respiration, this is this is in fact made possible by a continuous supply of O2 and the tissue by hemoglobin, is it? So the cultured cells, basically cultured cell, whatever cells that we culture, is it, the cultured cells, now we'll come back to our topic. The culture cell mostly rely on dissolved oxygen which is present in the media. Okay, which is present in the media, and that may be toxic at high concentration, because the oxygens are ultimately, when it reacts with some radicals, they will produce some acidic components, is it, free radicals, and that leads to toxicity generation, that leads to acid generation. Therefore it is very necessary to supply adequate quantity of oxygen, so that the cellular requirements, like that, met in a proper amount. So oxygen is not that much of we need to provide it in the external way, but the if you are if you are continuing your laboratory culture or tissue culture in a laboratory grade and a small scale, you don't need the oxygen support. But if you are producing some high protein component in a large amount in the large scale manufacturing process, then ultimately you need a little bit supply of oxygen in compare with CO2 also. Okay, then again the temperature, you all know, almost our animal cells grow at 37 degrees centigrade. We are warm blooded animals, we need 37 degrees centigrade. So of course cells need 30 degrees and 37 degrees centigrade. Plus minus 0.5 is okay, it's okay, we can maintain that. But 37 degrees centigrade plus minus 0.5 degrees centigrade is the ambient temperature. Then if we talk in terms of osmolarity, the general, the osmolarity is the most of the cultured cells is in the range of 260 to 320. What is the unit of osmolarity, if you so, it's the MOSM. Okay, MOSM per kg, that is the unit. Okay, so human blood plasma osmolarity is 290, remember that term. So we need to regulate in terms of osmolarity. Okay, next one is the, we need to study that what is the content, like what is the natural any media contents are there. The first of all, the buffering system should be there, like sodium bicarbonate buffer system. Then if we can use the HEPAS also, is it, then we can use phenol red as a pH indicator. It's the good pH indicator. When it reacts with some radicals or when it reacts with the um, proton components, it leads to change in the color and leads to become a yellowish, more yellowish, orange to yellow it turns. Okay, yellow is the highly acidic, and when it reacts with some hydroxyl ions or salt solutions, it becomes more violet. First of all it turns it into violet, and then ultimately it turns it into bluish color. So wide range of pH we can observe optically, so that's why phenol red is the good indicator. Then inorganic salts are there, amino acids are there. Out of that, L-glutamine is very important amino acid, so it provides the good protein buildup of the cellular mass. Then the carbohydrate, proteins and peptides are there, the several like albumin, transferrin, aproteitin, phetuin or fibronectin. These are the several protein molecule that we need to understand. Fatty acids and lipids are there, vitamins are there, and trace elements are there. So these are particular few components of our basic cell culture media. Then we'll see what different common cell culture media is right now available, right? uh, if you see, if you see the common culture media, then we generally come up with EM, which has been previously used, Eagles Modi minimum essential medium. Then the other media comes up with the Dalbecko's modified essential medium. Many industries provides Dalbecko's modified Eagles medium with a different variety, like Gipco or Sigma Aldrich or even Biorad. So these are other companies, or the even though High Media is also providing a good qualities of these media, and this media comes up with the low glucose, like high glucose and low glucose content. Then it also comes up with other like other component or other um, factors, like presence of glutamine, absence of glutamine, presence of particular growth factor, absence of that particular growth factor. So these are all the media that comes up with a variety of different components. We need to choose according to our study or according to our experimentation. RPMI 1640 is the very important media. Again, we'll see in details in our next lecture what is RPMI basically and why it is used as randomly in a high heavy level. Hams nutrient mixture, DMEM, F12 medium. So these F12 and DMEM and Hams, these are basically useful in the stem cells culture. Then ISCO's modified Delbecko's medium, IMDM. This is also one of the medium, and currently many researchers are using this IMDM media. So criteria for selecting the media, based on what we generally select. First of all, the intermediate survival, prolonged survival, indefinite growth, specialized functions. These four qualities, and based on this, like what is the survival rate, and few cell lines survive in a particular media for a long period of time, few cells survive in such a media in a short period of time, they exhaust it immediately. Somewhere they got a good amount of nutritional media and consistently they got a required amount of nutritional factors, and somewhere they don't get a required amount of nutritional factors, so ultimately they lose their activity of development or growth, and so ultimately they die immediately. So there is like indefinite growth. The specialized functions, few growth factors shows a very good specialized functions which leads to cellular attachment with the adhesion or addition to something, okay, in organ culture. So based on these criteria we generally select animal tissue culture media. It's always good to start with the MEM for adherent cells and RPMI 1640 for suspension culture, is it? So this is the text, or this is the wide range of cell lines, their morphology, the species from they have taken, and the media which has been used to culture these cell lines, and their applications. So I hope so. Uh, we have covered a little bit topic about tissue culture media. We need to cover much more about the media, but we'll see in the next lecture. Up till now you just write down this chart because this is very important. Just pause the video and write down it in your textbook. These are very important, and we'll see in the next lecture. We'll come up with the serum containing media, the specialty of serum containing media, and serum free media. Before that, we'll see the major constituents of serum in detail. Okay, chalo, take care, bye bye.